So good morning, everyone. We're going to get started. Welcome to this session called Topical Agents for PrEP, as good as systemic PrEP. I'm Rafi Landovitz from the University of California, Los Angeles, Center for Clinical AIDS Research and Education, and on behalf of my co-moderator, Jared Baton from the University of Washington, welcome to the session. We're glad you're joining us. Some brief opening remarks, and then we're going to go into a really Hollywood A-list set of discussions from our panel who are sitting up here, and then we will have about 30 minutes at the end for questions. So I'm going to ask that everyone please hold your questions until the end, and then we can hopefully engage in a broad discussion at that time. So I'm sure no one will forget the extraordinary excitement and electricity in the room in Vienna in 2010 when Slim and Quraish Abdul Karim presented the results of the CAPRISA 004 study, really demonstrating for the first time proof of principle for a topical antiretroviral agent as pre-exposure prophylaxis. Of course, the science paper that was published at the same time lives on as a critical reference to anyone involved in the PrEP field. Of course, later that year, as Sheena McCormick already schooled us this morning, Bob Grant released the results of the iPrEP study demonstrating an initial 44% reduction in HIV acquisition in individuals randomized to TDF-FTC in the placebo-controlled iPrEP study. And my co-moderator for this session, Jared Beaton and his colleague Connie Kellum and others from the Partners PrEP study, of course, presented the staggering and behemoth results of the Partners PrEP study, demonstrating up to 75% reduction in HIV incidence among heterosexual men and women in serodiscordant relationships, also published in the New England Journal of Medicine. Of course, the field was turned on its head when Jeannie Marazzo presented the results of the VOICE trial, which was supposed to be the confirmatory follow-up study, particularly for HIV uninfected women, regarding topical, albeit daily topical, tenofovir gel use, along with TDF and TDF-FTC in that population, really sending both providers, consumers, public health practitioners and advocates struggling to really understand the differences between different populations and why discrepant estimates of efficacy and effectiveness were coming out of these studies. The 2015 CROI found Sheena McCormick demonstrating in the Pragmatic Proud study 86% reduction for PrEP use in the immediate setting as opposed to deferred setting in the UK. And of course, at that same conference, in a really electric session, Jean-Michel Molina presenting the results of the IPERGAY study, also subsequently published in the New England Journal of Medicine, with on-demand TDF-FTC compared to placebo having a similar reduction in HIV incidence among MSM in France and Canada. The confirmatory randomized study for topical tenofovir gel, of course, was presented by Helen Rees last year at the CROI conference, and the disappointing negative results probably are in the process of being further explicated with some detailed analyses, and I believe are under review currently at a major journal, and hopefully we'll be seeing those soon. Of course, the sine qua non of all of this seems to be that adherence seems to be extremely closely correlated with observed effectiveness. But there may be other factors that are contributing to these results, as we're going to hear from our esteemed speakers today. And I would leave you by reminding you that Beyonce did remember everything and get it right when Jared and Annalene presented the results of statistically significant efficacy for the Dapivirine rings, again with use-dependent evidence of effectiveness, and open-label studies are ongoing. So I think as we consider the questions that our panel is going to attempt to address today, I would provide you with the following context. Do we have the right compounds? First, do we fully understand the potential of the existing compounds that are available from the existing placebo-controlled studies? Can we get the products to their right anatomic locations? Can we protect all the right places from a single compartment topical application? Will the right people use it enough, implication for it to work? And is there the political will and infrastructure to bring them to scale? And I have political asterisk or starred there to acknowledge the questionable role of the previous leader of U.S. funding to continue into the future to support this incredible, important work in collaboration with a number of other partners. And with that, I'm going to turn the podium over to our first speaker, who is Angela Kashuba from University of North Carolina. Angela is the John A. and Deborah S. McNeil Jr. Distinguished Professor in the Eshelman School of Pharmacy at the University of North Carolina at Chapel Hill. She's the chair of the Division of Pharmacotherapy and Experimental Therapeutics in the Eshelman School of Pharmacy and the director of the University of North Carolina CPHAR Clinical Pharmacology and Analytic Chemistry Core. She's an adjunct professor of medicine in the Division of Infectious Diseases in the UNC School of Medicine and is board certified by the American Board of Clinical Pharmacology. Her laboratory focuses on elucidating mechanisms of drug distribution in tissues and predicting efficacy. So Angela will be speaking to us today on Does Pharmacology Support Topical PrEP? Welcome, Angela.
Terrific. Thank you so much. It's very exciting to be part of the session, and I'd like to thank the organizers for their kind invitation. Okay, I guess I do that. These are my disclosures. And the goals of pharmacology are really to get the right drugs to the right site at the right concentration for the right length of time. And that goes for any therapeutic area. And so these are the tenets on which I'm going to base my talk. And I'm not going to focus on the right drugs per se, but I'm going to talk about the data that we have and the drugs that have been developed in terms of whether they get to the right site at the right concentration for the right length of time. So I'd like to start by looking at the right site and presenting some data from Craig Hendrix's group, which has used SPECT-CT to understand distribution of virus in the mucosal tissues of the female genital tract and colorectum, and also whether gel gets to those same sites. So he used virus-sized particles to place into the female genital tract and into the rectum and watch the movement of the particles through these spaces. And here you can see in blue that there are particles throughout the lower genital tract, and especially in yellow are high concentrations around the cervix and in the fornices. If you look at the gel distribution, you also see gel distribution similar to that of where these virus-sized particles move, and that is the highest concentration around the cervix and the fornices, and lower concentration in the lower vagina 24 hours post-dose. Similarly, in colorectal tissue, we see these particles moving throughout the lower gastrointestinal tract, and in some cases up to the splenic flexure. And 24 hours after gel administration, we also see extensive gel distribution. So in this set of experiments, he found that the gel does cover the physical location of where virus-sized particles move. We don't have the exact same data for the rings, but we do have some very nice PK data that may help us understand this. So this is a concentration time plot of mean Dapivirine concentrations over days, and there are concentrations, mucosal fluid concentrations attained at the placement of the ring, at the cervix, and then at the lower vagina. And so you can see that the cervical concentrations are also high, lower concentrations in the lower vagina. But does drug actually get to the site of infection? So we see that drug kind of covers the areas where a virus may move, but does it get to the site of infection? Well, what is the site of infection? That's a question that we've had in this field. This is a cartoon of the early events in HIV infection, where HIV is seen crossing the epithelial barrier, potentially being caught up by dendritic cells and finding their way to activated CD4 positive T cells where there is infection, dissemination of virus, and establishment of a lymphoid tissue reservoir. We think the stroma is a likely target to abort HIV infection. There's been some data in the literature suggesting that there might be trafficking of virus by dendritic cells, but this appears to perhaps be more rare. So we're going to focus on the stroma and see if topicals really get here. Well, David Katz from Duke has presented some very nice data on Raman spectroscopy and understanding drug concentrations after tenofovir gel application throughout mucosal tissue. This is a cross-section picture of mucosal tissue with about 100 microns of epithelium and then about 300 microns of stroma. And if you plot tenofovir concentration compared to depth of tissue, an hour after administration of tenofovir gel, this is the concentration gradient that you see. So the concentration in the stroma is only about 20% of what we see at the epithelial surface. 2 hours later, we see less of a concentration gradient and not much accumulation in the stroma, still about 20%. We don't have the same kind of Raman spectroscopy data for rings, but we do have some good information in the mucosa of sheep in which a Dapivirine ring was placed for 28 days. The tissue was removed, frozen, slices were cryostated and measured by LCM-SMS. And so you can see, starting at about 300 microns, you see that there is also a decline in concentration across the tissue. So the question now is, are these concentrations adequate for efficacy? Well, for topical gel, for tenofovir in particular, I'm going to focus on tenofovir diphosphate for a moment, because tenofovir is not the active form of the drug. It has to get into the cell and be phosphorylated into the diphosphate form of the drug. So what I'm presenting for you here is tenofovir diphosphate concentrations versus time post-dose of a gel, either a single dose or multiple doses. What I have here for you in red is the PBMC concentration of tenofovir diphosphate at about 100 femtomoles per million cells that is found after you give an oral dose of tenofovir, so that's sort of going to be our benchmark. So if you look at the concentrations in pink here, these are endocervical cell diphosphate concentrations, and they're very high, 100,000 femtomoles per million cells. Concentrations in vaginal tissue are a bit lower. These are 100 to maybe the low thousands of femtomoles per million cells. And then if we looked at rectal tissue after rectal gel administration, we see that those concentrations are also in the same range as vaginal tissue concentrations. So the question is, are the lowest of these concentrations in the hundreds to thousands adequate for efficacy? Well, we have some data from the CDC to try and address this issue, where they gave a dose of tenofovir gel, and then they measured vaginal lymphocyte concentrations 30 minutes after a dose here. Those concentrations were in the thousands. When animals were challenged 30 minutes after a dose, they had 100% protection. And then they measured concentrations all the way to 72 hours after a dose of gel, and they were in the hundreds. When they challenged animals at this time point, there was about 70% protection. So absolutely we see that there is a relationship and evidence of gel efficacy from a pharmacologic standpoint, although perhaps not at 100% if our concentrations are at, in the hundreds rather than in the thousands. Now let's look at the clinical correlates of this. These are some data from Caprisa 004, where tenofovir 1% gel was given in a coitally dependent manner to women, and this is a case control analysis investigating the percent infected on the y-axis versus the concentration of tenofovir in the vagina on the x-axis. And you can see that as the concentration of tenofovir increases, the percent infected decreases, although these are small numbers. And we have a cutoff of about 50% here at 1,000 nanograms per mil in terms of tenofovir concentration in the vagina. And this equates to about 200 femtomoles per million cells of tenofovir diphosphate in the vagina. So in the hundreds, we're also seeing efficacy. We have some similar data with HSV-2 infection, where our cutoff seems to be 10,000, and this concentration of 10,000 nicely aligns with concentrations that we see in tissue challenge models, where the EC50 is about 10,000 nanograms per mil. So there's definitely a concentration effect relationship of tenofovir with HIV protection and HSV-2 protection. Well, what about rings? Here are some data from Smith et al., published a number of years ago, evaluating pigtailed macaque vaginal tissue concentrations and cervical tissue concentrations 14 days after TDF intravaginal ring. They measured lymphocyte concentrations in the proximal vagina, the distal vagina, and the cervix, and for this particular ring, they also found concentrations in the thousands. I'm showing you this PK curve one more time because I like it so much, with Dapivirine concentrations over time. And you can see that the fluid concentrations are much higher than the IC90 of about 10 micromolar. We know that tissue concentrations are about a log lower for Dapivirine than fluid concentrations. So if we take these concentrations and move them down by about a log, we still see that the concentrations are right around the IC90. If infection is occurring in the lower vagina, potentially there might be some limitations here, but in general, from pharmacologic principles, we should see efficacy. And finally, the right time. There's been discussion about how long to protect tissues after exposure. What should the concentrations be after the last dose, and how do we continue to protect mucosal tissue? Well, we don't really know exactly what that time period is in vivo, but we do know in vitro that replication-competent HIV can reside in tissue cultures for up to 8 days. So we think maybe a 7-day period of time is important. Well, if we look at some data from CAPRISA again, in a group of highly adherent women using the gel, we plotted their concentrations in the vaginal cavity compared to the time after their self-reported last dose. And we see that early on, we do have prolonged tenofovir exposure, and we have a half-life of about 2 days. In addition, the diphosphate in tissues or in cells also has a long half-life, so we think that there is indeed protection here after a dose. And clearly for the Dapivirine ring, there is sustained concentrations. The only problem is after the ring is removed, where fluid concentrations drop dramatically within 2 to 3 days, and subsequent tissue concentrations would also be low. So it would be important for women not to remove the ring anytime close to when a potential exposure has occurred. So does pharmacology support topical PrEP? I think yes. We've gone through the right site, the right concentration, and the right time. If the formulation allows distribution to the viral locations, if the active compound can penetrate deep into the mucosal tissues, if the drug achieves potent concentrations at these sites, if the metabolite has a long residence time to cover the virus, although there are some caveats to this. Is there virus trafficking to other sites where topical administration may not reach? So the more drug you give, you're just not getting the same amount of drug at a distal site, and that's considered a ceiling effect. There might be other physiologic processes that also interfere with the PK-PD relationship, like inflammation. We've also heard a lot about the microbiome recently, and I think you're going to hear more about that in the next presentation. So I'd like to thank you for your attention.
Thank you, Angela. Our next speaker is Dr. Slim Abdul Karim. Dr. Karim is a South African clinical infectious disease epidemiologist who is widely recognized for his research contributions in HIV prevention and treatment. He's the director of the Center for the AIDS Program of Research in South Africa, or CAPRISA, pro-vice chancellor of research at the University of KwaZulu-Natal in South Africa, and professor of clinical epidemiology at Columbia University in New York. He's the chair of the UNAIDS Scientific Expert Panel and serves on the Gates Foundation Global Health Scientific Advisory Board, the WHO HIV-TB Task Force, and the WHO Expert Panel on Sexually Transmitted Infections and HIV. Slim will be speaking to us this morning on lessons learned from tenofovir vaginal gel studies.
Thank you very much, Rafi. And indeed, it's a great pleasure to be with you here this morning. I want to thank Jean-Michel Molina for putting this session together. And I hope you'll indulge me and allow me to acknowledge some of the stalwarts we have in the room. And while I'm doing that, I just want to tell people standing at the back, there's a whole lot of seats in the front if you want to take this opportunity to come and take up the seats in the front. So as I was walking in, sort of a bit of nostalgia in looking at the stalwarts. And stalwarts, I mean people who've been at this field for more than 20 years, when you go back to 1997. So I want to acknowledge 5 of them. There are many more in the room, but just 5 of them. So to start with was Ken Mayer, who's actually grown a beard now, so that he's looking more like a stalwart than he did before. The second is Zita Rosenberg. As I walked in, I saw her sitting at the back. And Gustavo Doncel, who is hanging around somewhere in the middle of the audience. And then I saw Sharon Hillier arrive in the room. And not just Sharon, but with Butch as well. And Sharon's really one of the doyens of our field. And finally, Connie Kellum, who's joining us up here in front. So just a big thank you to all of those people who really defined our field and helped us move it forward. So I'm going to talk to you about lessons learned from the research on vaginal gels. Okay, so let's see. Let's get this moving. Okay, let me press here. So I'm going to briefly talk about the evidence from the trials, from the 3 trials, and I'm going to just concentrate on 4 lessons. Adherence, the role of the microbiome and its importance, talk about genital inflammation, and then end off with some comments on multi-purpose technologies before making some concluding remarks. So when we look at the current evidence, the intent to treat analyses from the 3 tenofovir gel trials, the Caprice 04 trial, the Voice trial, and the FAX trial, this is the range of efficacy that we see, from 0% all the way through to 39%. But if we do a slightly different analysis, if we look at the overall intent to treat, and now we do a case control analysis comparing the adherent individuals within the active arm, then we see a little bit more consistency across the 3 studies, where across the Voice study, the Caprice study, and the FAX study, we see efficacy ranging from 52 to 57%. Now, this is not the overall clinical trial analysis. It is a subgroup analysis, and it's an analysis that's intended to give us some impression of what impact adherence had. But for me, the message that it sends is that adherence is essential, and that if you adhere, you have the potential to benefit from the gel. And we saw this in different ways. In the Caprice 04 trial, when we stratified and looked at the overall analysis across the placebo arm and the active arm and compared the 2, looking only among adherent individuals, we see much higher level of protection. And one of the important things to feature in adherence, as we've noted, is that as we design these studies, we have to design them with women in mind. We have to consult them. We have to factor in the issues that they most have to grapple with as they use the products that we are designing for these studies. So the first issue is the importance of adherence. And adherence becomes even more important as we think about the role of the microbiome. And in these data, in a collaboration that we undertook with the Public Health Agency of Canada and the University of Washington in Seattle, we divided and we stratified the data for women who had lactobacilli dominance and those who had greater diversity. And what we showed, let me just go back. What we showed is a higher level of effectiveness among women that had lactobacilli dominance. So that raised the question, so what's going on with these other organisms? The dominant of the other organisms is Gardnerella. And what we found was that Gardnerella, in the presence of Gardnerella, the levels of tenofovir were rapidly declining. And we saw that it was not just the overall tenofovir levels in the supernate, but it was also in the diphosphate formulation of the drug. So this told us that the importance of the microbiome, that we have to factor in that it's not just the host and the drug, it's also the host drug and the host microbiome that features in the pharmacokinetics. The third lesson we learned was the importance of genital inflammation. And much of this genital inflammation is not what we traditionally think about in sexually transmitted infections. Indeed, as we think about STIs, we think about gonorrhea and we think about vaginal discharge and so on. Much of the genital inflammation we have seen in the studies we've been doing is a more asymptomatic form where we can identify these women through the presence of raised inflammatory cytokines. And what we show is that inflammation not only increases risk of HIV, it also impacts on the efficacy. So when we look at among those who are adherent and those who are not adherent, we see that almost if you have genital inflammation, it's almost like you're not taking the drug. And so that made us think about, so what is it that genital inflammation is doing? Well, there are two things we identified. The first is that when we look at the infectivity of the viruses, among women who do not have genital inflammation, most of the viruses that are causing infection are those that have high infectivity index. You know, they're able to cause infection because they are high infectivity. However, in women who have genital inflammation, we see a large proportion of low infectivity virus. So the threshold for which viruses cause infection changes as a result of genital inflammation. The other thing we found, which I'm not going to present in great detail, is that we see CD4 cell numbers increase in the vaginal mucosa, and we also see that they are closer to the surface. And the last lesson I want to focus on is just a reminder that as we think about topical agents, we're not just thinking about preventing HIV, but we're thinking about all of the other things that we need to prevent as well, whether they are sexually transmitted infections or pregnancy. And if we can combine all of these and achieve them in a single formulation, all the better. So let me end off by just reminding you that the challenge that we continue to face today of incredibly high rates of HIV in young women in Africa, and these are HIV prevalence rates from pregnant women at the 7 clinics that they attend in our community of Vulindlela where we do our research. It's a stark reminder of the challenge that we face every day in dealing with the HIV epidemic, and that for these women, we have to do as much as we can in order to make a range of options available to them, whether they are in the form of rings or gels or tablets or any of those. So let me end off with saying that what we've learned, well, tenofovir gel prevents HIV and HSV2 in women. We know that the trial outcomes vary from 0 to modest, and we know that adherence was a major challenge explaining those. Adherence by itself becomes even bigger, a bigger problem in the presence of a microbiome that is not lactobacilli dominant. And future PrEP trials are going to need to factor in the effects of genital inflammation and the vaginal microbiome as they think about moving forward. And women want a range of prevention options. While tenofovir gel could contribute to HIV prevention, the higher priority, as I see it, is a long-acting formulation that reduces the burden of adherence, especially on young women. Let me just end by thanking many of our collaborators who have been important in contributing to the data I've presented today. Thank you very much.
Thank you, Slim. That was fantastic. And to reiterate what Rafi said at the beginning, not only do we have a group of A-listers talking today, we also have some of the most dedicated and wonderful people, I would say, working in HIV up here today, including our next, who's Annalene Nell, who is Chief Medical Officer and Executive Vice President at IPM. She has led IPM's clinical program for the Dapivirine ring, culminating in what we'll be hearing about today. And we're very excited to hear what you have to talk about, Annalene. Thank you.
Thank you, Jared. Thank you for the opportunity to discuss intravaginal rings for PrEP. What are the future prospects? And I will discuss women's vulnerability to HIV, providing multiple options, and then focus on the Dapivirine vaginal ring, as well as the current regulatory pathway, as well, including the next steps, and then future prospects of possible intravaginal rings and combination products. Women and girls are particularly vulnerable to HIV acquisition due to economic, biological, and social cultural realities that we were reminded of over the past days. Physiologically, women are more likely than men to acquire HIV during heterosexual intercourse, and young girls are most at risk. In sub-Saharan Africa, the number of new HIV infections amongst adolescent girls and young women aged 15 to 24 years remains exceptionally high, as we've heard, with approximately 7,000 new infections per week. These young women are at least twice as likely to be infected with HIV as their male partners. Given the continuing high HIV infection rates among women in sub-Saharan Africa, microbicides are needed for a comprehensive response to HIV and AIDS, as current prevention strategies don't offer women adequate self-initiated options to protect themselves. Ending the epidemic will require different and multiple prevention options to meet women's individual needs, since no one option will satisfy every woman. IPM holds an exclusive worldwide license for Dapivirine from Janssen's Science Island, part of Janssen's pharmaceutical companies of Johnson & Johnson. Dapivirine, also known as TMC-120, belongs to a class IRVs called non-nucleoside reverse transcriptase inhibitors that bind to and disabled HIV reverse transcriptase enzyme. The ring was developed by the International Partnership for Microbicide and designed to ensure that women in low-resource settings have cost-effective options and access to any Dapivirine-based microbicide. The Dapivirine rings is made of flexible silicone, and women can insert and remove it themselves. It is placed inside the vagina close to the cervix, where it slowly releases the active drug Dapivirine over the course of a month. Two independent large-scale phase 3 trials demonstrated that IPM's monthly Dapivirine vaginal ring has a favorable safety profile and provides a statistically significant reduction of the overall risk of HIV infection by approximately 30% compared to a placebo. That can make a significant impact on a community where the norm is up to 9% incidence. Across these two trials, it was demonstrated that in participants who used the ring at least some of their time, their HIV infection risk reduced by 45%. The ring showed no HIV risk reduction in women under 21 overall, likely due to low product use. Additional research is underway to better understand barriers to ring use among the youngest women and to support adherence in this high-risk group. A similar proportion of women in both groups had NNRTI mutation at HIV-1 seroconversion. Additional phenotyping and deep sequencing results are expected in future. Supportive trials also showed that the ring does not impact the function of the male and female condom. IPM is seeking scientific opinion via Article 58 from EMA of the Dapivirine ring for women ages 18 to 45. This was the same age group that participated in the Phase 3 safety and efficacy trials. The dossier was submitted on the 22nd of June 2017, and I want to acknowledge each and every one that directly and indirectly supported us to reach that important milestone. The evaluation of medicinal products for human use under Article 58 allows EMA to give scientific opinions in collaboration with WHO on products that are intended exclusively for markets outside the EU, thereby increasing access to medicines and improving public health in low and middle income countries. A positive opinion from the EMA will be used to facilitate WHO pre-qualification, thus facilitating the submissions and approval in target African countries, including Kenya, Malawi, Rwanda, Tanzania, Uganda, and Zimbabwe. These countries all participated and contributed to the Phase 2 and Phase 3 program of the Dapivirine ring. The next regulatory step for IPM is to file to the MCC, the Medicine Control Council in South Africa, in Q4 2017. It is a high priority for IPM given the higher HIV incidence rates among women in these countries, and especially South Africa. In addition, IPM will seek regulatory approval from the US Food and Drug Administration. Submission to the FDA is targeted for 2018. In any given month during the phase 3 trials, 75% of women demonstrated some ring use. As with other HIV prevention clinical trials of investigational products, the women were not only using a vaginal ring for the first time, but also had no knowledge of its efficacy or long-term safety, or whether they were receiving an active ring. The open-label trials now underway will give a closer indication of real-world use while obtaining more long-term safety data. Additional research to better understand how young women use the Dapivirine ring and ways to support consistent use among the high-risk group is underway. This includes a study to begin later this year called REACH, led by the Microbicide Trial Network, which will assess use of both the monthly Dapivirine ring and daily oral PrEP among young women ages 16 to 21 years in Kenya, South Africa, Uganda, and Zimbabwe, looking at both product safety and young women's preferences for each. In addition, IPM is adding to its Dream Ulay a cohort of 600 young women ages 18 to 25 who will be using the ring for the first time, which will provide important insights into product use and more effective adherence support strategies. 2 additional clinical trials, one in pregnant women and one in breastfeeding women in sub-Saharan Africa, are planned by the MTN to commence in 2018. Given the continuing high HIV infection rates among women, currently available prevention options have not done enough to stop the spread of the HIV epidemic. More women-focused HIV prevention choices and additional multi-purpose prevention options are critical to reduce the burden of HIV infection in our countries, but also to enhance women's health and meet women's individual needs. Long-acting and on-demand products could be more cost-effective, convenient and acceptable to women, especially if multiple transition pathways are covered. Therefore, there are currently various products in development by product development groups and partners, and many of you are in this room, and I want to say thank you. The focus is to have products that prevent HIV infection via various mechanisms of action to provide a higher HIV infection risk reduction and reduces the chance to possible resistance. But also other combination products are under development. For example, HIV and pregnancy, combination preventing STDs, and longer-acting products, for example, 3-monthly rings. As the only current long-acting HIV prevention method, the Dapivirine ring, if approved, it could have an important public health impact as part of a comprehensive HIV prevention package. The advantages for future long-acting and multiple prevention ring options are it can deliver drug directly to the local site of potential infection to minimize systemic exposure and side effects. Women can self-administer the ring, discreetly use it, and it can reduce the burden of adherence to product use, especially 3-monthly rings. A vaginal ring is not familiar in the African setting, unlike Europe and USA, where there is familiarity with the contraceptive rings. Demonstration and implementing programs will be important for success, and the collaboration between us will continue. More information will be shared with regards to the Dapivirine vaginal ring during all these listed sessions. The data of the pooled efficacy data on breast milk, adolescent females, as well as the effect of menses and tampon use on the pharmacokinetics of the Dapivirine vaginal ring. As you can see, it took a true village of people to get the first ring as far as it is at the moment. Thank you very much.
Thank you, Annalene. What a great history. Our next speaker is Ian McGowan, who's professor of medicine at the University of Pittsburgh, trained at Oxford and at Liverpool. He really has led the idea of rectal microbicides for HIV prevention, and is one of those few people who can really think in clinical science, in basic science, in behavioral science, and in community for implementing something new like this. So thank you, Ian.
Thank you very much, Jared. Good morning, everyone. It's a pleasure to be here. I think everyone in the room would acknowledge that both men and women are at risk of HIV infection through condomless anal sex. I think the question I've been asked to address is really the question of rectal gels for pre-exposure prophylaxis. Are they an option? I emphasize I was given that title rather than chosen it, so it will be interesting. What am I going to do over the next 10 minutes? I'll tell you, for those who don't know, what rectal microbicides are. Perhaps ask, are they needed in 2017? Where is the science? What are the obstacles to the development of a safe and effective rectal microbicide? And end up where we begin. Rectal gels, are they an option? So what are rectal microbicides? Well, they're topical products which are designed to prevent or significantly reduce the acquisition of sexually transmitted diseases, including HIV infection. Currently, in the various stages of development, we have gels, we have enemas, and we have rectal inserts. They've been postulated to be used as a daily product, daily administration, or perhaps more likely as event-driven products. And the majority in the pipeline are antiretroviral, although there are some non-classical antiretroviral products being developed too. Just in terms of formulations, here we have a gel, as you can see. Craig Hendricks is leading a very innovative approach using tenofovir enemas to deliver the product. And Conrad have, as you are aware, inserts or suppositories, whatever you want to call them, as another potential solid dose formulation. So, are they needed? Well, it's been already mentioned this morning that people like choices. I think we can agree upon that. Some people indeed may prefer a gel, a ring, an injection, an oral tablet. I would argue, and still argue, and I see Alex is in the room, who's pioneered much of this work, topical products are behaviorally congruent. People use gels, enemas, associated with anal sex. This might be a good way to deliver the product. And I think it is an ideal approach for event-driven PrEP. And I think we have to acknowledge, across the spectrum of prevention, no PrEP agent or formulation has a perfect target product profile. There's always going to be some issues, some gaps, which will probably lead to discrimination in terms of choice and preference. So that's all the good stuff. What else? Oral PrEP, thanks to the work of many people in this room, is now licensed, safe, highly effective, and increasingly available. That's a reality in 2017. Dual compartment protection is going to require systemic administration of PrEP agents unless the topical product is used both rectally and vaginally. We have no PK data suggesting that if you use a vaginal gel, you will see PK exposure congruent with protection in the rectal compartment. And again, perhaps stating the obvious, public health and research resources are limited. They're finite. So where's the science, very briefly? Well, we have very good non-clinical and animal model data to support both the safety and effectiveness of topical PrEP agents. I'm not going to go into detail, but certainly in the humanized mouse model, in the non-human primate model, we've seen good protection with a number of agents. As a consequence, many products have advanced through phase 1 evaluation, generating safety, acceptability, and PK/PD data. In fact, with respect to tenofovir gel, this has been evaluated in a multi-center international study, the MTN 017 study. But as of yet, there have been no phase 2b/3 effectiveness studies conducted, and that's clearly an issue. So, I think Angela's touched on this very briefly. Will this approach work? Whenever we talk about feasibility, we always steal Craig Hendricks's beautiful imaging slides. This is basically the same kind of presentation that Angela showed. It's fused CT SPECT imaging. This happens to be a HIV surrogate in the rectal vault. And in this particular individual, on this occasion, you can see the distribution of the gel. So I think at a macro level, you can deliver gel, which will indeed travel to the regions that you might anticipate could be sites of infection. I think at a micro level, it's a little more contentious. And there's some very nice work, and certainly in the mouse model, looking at how far will a gel actually extend to provide comprehensive coverage of rectal crypts. So if you will, at the histopathological level, there may be more challenges achieving that goal. This is just some work from a study I did in collaboration with others, the CHARM-1 study. It was an evaluation of 3 different formulations of tenofovir gel. What you're seeing here is an explant infection, PK/PD plot. On the y-axis, you can see the amount of virus in the tissue. On the x-axis, the amount of drug. And you see this inverse significant correlation, more drug, less virus. So if you will, I think that's a clue to potential efficacy in the real world. So what are some of the obstacles? Scientific, first of all. As I've said, mucosal coverage, comprehensive at a histopathological level, and even within the tissue may be problematic. Topical products can certainly provide tissue protection, drug levels, but will they provide the draining lymph nodes of the level of drug that might be needed to abort infection? That remains a relatively understudied and important question. As I've said, men and women are at risk of HIV infection, and a single topical agent may not provide dual protection, which is put here. What about consumer preference? This is just another big issue. This is what we've been told over the last decade or more. This is what the consumers want. They want a sexy, lubricant-like product which protects them. And as of yet, in 2017, this is what they get. And that really hasn't set the world on fire, as you can imagine. And we don't have the resources to produce anything better. This is really a commercial issue. But that's a challenge when you say, hey, how about a rectal microbicide? Do you like this? Not so much. So, regulatory issues quickly. With the increasing availability of oral PrEP, very high levels of efficacy, almost 100%, we no longer have the ability to conduct classical placebo-controlled trials. Alternative design strategies are going to have to include methodologies such as non-inferiority trial designs, and these can generate very large sample sizes. But, Rafi, as an example here, they managed to contain in their study design for cabotegravir a sample size of 4,500, which is doable, but I wonder if that will be repeated in other studies. And I really encourage you to attend this session, which I think is really addressing how do we move forward with effectiveness trials of new PrEP agents. It's a big problem. Programmatic. Oral PrEP is increasingly available. The second generation of oral PrEP, FTC TAF, is being funded by Gilead Sciences, 5,400 people. So industry is finally engaging in funding directly PrEP research. We have a ring, you've heard about it today, positioned, indeed, undergoing regulatory review and potential licensure. And we have a very large cabotegravir study up and running, data anticipated at the end of 21. Do we really need anything else? That's a question which a number of people are indeed asking. As some of you may know, there is a robust dialogue ongoing now about the future of the NIH-funded clinical trial networks and what is actually needed. And this is just a slide taken from a presentation the director of DAIDS, Carl Diefenbach, gave earlier this year. And I just highlight what he felt was needed for the biomedical pipeline. Safe, desirable, durable, and affording systemic protection. Durable and systemic protection are not two phrases associated with topical agents. They're event-driven, and they're probably not going to act systemically. So I think at this level, at least, there isn't huge encouragement that there will be large amounts of federal funding for a phase 3 study of a rectal microbicide. So I'm going to end with my last slide, the Mona Lisa. Everyone always wonders what she's thinking, and I think she's probably thinking about rectal gels. And are they an option? And I'm not going to tell you what my answer is. Thank you very much.
there's no easy transition from that. Our last speaker is Jessica Haber, who is Associate Professor of Medicine at Harvard Medical School and Director of Research at the Massachusetts General Hospital Center for Global Health. I think there's no better introduction than what someone told me today. She is the queen of adherence, and certainly the queen of adherence related to PrEP. So we're really happy to have her here.
Okay, good morning. Thank you very much to the conference organizers for the opportunity to speak with you today. And unlike my predecessor, I didn't like the title, so I changed it. Because when I was thinking about what I wanted to say to you in the next few minutes about adherence, I didn't want to just focus on one or two agents or one or two properties of agents. So I kind of summed it all up together, and we'll talk about topicals in relation to other types of PrEP. So I am funded, but I do not have any conflicts. And so what I'm going to cover in the next 10 minutes, we'll start by thinking broadly about what are the factors that influence adherence. I think we've heard from numerous speakers today and from many aspects of PrEP that adherence is really important, but how do we think about that, and how do we come towards some interventions and ways to help people with it? And acceptability is wrapped up in that. Talking a little bit about multi-purpose technologies, as you've already heard about some today. And a concept of prevention effective adherence, so how can we help people adhere when they need it most? And then a little bit of perspective on adherence and effectiveness as PrEP is evolving over time. So I know this slide is very busy up here. This is taken from one of the conference organizers, Linda Gale Becker's publication in The Lancet a couple of years ago. And I don't expect you to think about all these different areas and all the different, very small print at the bottom there. But what this slide serves to do is to think about adherence broadly. So we can't just think about the individual or the product or their environment, but really what are all of those pieces and how do they fit together? And what I'm going to focus on in the next few minutes is really just a couple pieces of that, but please keep in mind that there's a much broader context. So specifically, I'll be talking about the agents themselves and the individual behavior around those agents. So does this pointer work?
No, I think use the mouse.
Use the mouse, okay. Great, so this is, okay, here we go. So I broke up, as I mentioned, I'm not just talking about topicals, but I decided to break up adherence or PrEP products into types of use. So up here we have daily, which includes oral tablets and could include vaginal rectal microbicides. Then we have, this mouse isn't really working. And then periodic, which I'm thinking about vaginal rings, and then injections, and then on demand, which could be tablets or vaginal rectal microbicides. And there are many, many factors that could be facilitators for adherence or barriers, but the ones I've listed up here are kind of the unique ones to consider as we think about daily, periodic, or on-demand PrEP. So looking first at daily, how do people adhere to medications daily? Well, they tend to do, again, people do things in a lot of different ways, but one of the most common ways is to form a habit, whether that's pairing the PrEP with brushing your teeth or watching a show, something that becomes part of daily life. And for many people, that's a very successful strategy. But that can also be tiring. Sometimes people don't like brushing their teeth. Sometimes they may not want to take PrEP. And the more often you do something, the more potential there is for stigma. So there are pros and cons to taking a pill every day or using a product every day. If we move into periodic use, maybe that infrequent dosing is a benefit for someone who doesn't want to think about HIV protection every day. They want to have something that's more event-driven. And maybe there's less potential for stigma if you don't have to do this so often. But on the flip side, you have to then remember to take it. And if you're not doing that as part of an ingrained habit, how do you remember to do that on an infrequent basis? Then there may be some issues with discomfort, either physiologically or psychologically, with some of these formulations. If people don't like the injection, the way it feels, or they have an issue with the ring inside the vagina, these are some things that may not work for everybody. And then thinking about the final column there with on-demand, again, infrequent dosing could be a really good thing, especially if it is event-driven. So I think I misspoke earlier. The periodic ones are not event-driven. I was thinking of these. But being prepared for those cues is really potentially a challenge, and we'll talk about that a bit more in a moment. So also thinking about acceptability in sort of a framework way, This is a nice model that was published by Tully and colleagues a couple of years ago. We're again thinking about the products in the middle is where we're focusing today, but thinking about the individual, the skills and preferences of that individual, the community, the attitudes, the relationship context, and getting broader out to cultural norms and policies and legislation. All these factors really impinge on acceptability and how that impacts on adherence. So this is another slightly busy slide. There's an online survey that was published a year or so ago. This is from MSM in the US, but just showing that there are many different types of PrEP, as you've heard today, and people care. You know, they really don't all want the same thing. There is no one type of PrEP for each individual. So asking about it and having those options available to people is very important. And there have been a couple of nice satellite sessions. They've already happened, but you can go back and look at them online after the conference to hear more about individual choices and how people are thinking about those. And then moving into multi-purpose technologies, as we've already heard in other sessions, HIV prevention is important. It's certainly important to all of us. We wouldn't be here if it weren't. But not everybody walks around every day thinking, how can I prevent HIV today for myself? So if we can combine HIV prevention with other types of prevention, like against pregnancy or other STDs, as we've already heard today, this could be really, really important for adherence and acceptability. So the next area I'd like to move into is prevention effective adherence. And this is a concept that several of us have been developing over the last few years, which is a little bit different than the way we've thought about adherence for treatment. With a treatment adherence, you want to be taking medication every day for the rest of your life. There's really not a time when adherence isn't necessary. And in efficacy trials, the same thing can be said, that there's not really a time when non-adherence is okay. You really want to know if people are taking a medication or are using a product so that you can comment on efficacy. But life is not necessarily like that for PrEP. So are there times when people need PrEP and are there times when they don't? And how does adherence fit into that? So in this schematic that you can see here, we've very stylistically looked at behavior. So where we have adherence behavior here in those little boxes of taking PrEP, whichever kind it may be, people may take it for discrete periods. I've sometimes heard people talking about taking PrEP for life, and I don't think most people are going to be taking PrEP for life. There may be periods of sex work, or there may be periods of time when people are between partners or having multiple partners, but probably not for the rest of their life. So as long as HIV exposure is happening during those times when people are using some formulation of PrEP, then they will be covered. If you take it, you know, it works, right? But in those periods where you're not at risk, maybe don't take it. And as an adherence researcher, it's kind of like an anathema. You don't say it's okay to be non-adherent, right? But it is if you're not at risk. And so it's a bit of a paradigm shift and a different way of thinking, but I think we need to embrace it as we look at how to really help people take PrEP in whatever formulation they want to use. And this is really a way to be efficient. How do we get people protection when they need it? And that will minimize the burden, whether they're looking at daily, periodic, or on-demand use, and also thinking about rolling out PrEP. Something that needs to be used by people for really, really long times, those are quite expensive. But if we could think about more targeted use, that could increase the rollout of PrEP. So with prevention effective adherence, one of the key factors is knowing when you're at risk. And again, people aren't always walking around every day thinking, am I at risk for HIV today? So how can we help people do that? With daily and with periodic dosing, we should be thinking about seasons. So it isn't a daily question, but we need to know, is protection needed? It's much in the way that women often think about contraceptives. Do I need to be taking a pill for several months at a time? Do I want an IUD for the next few years? These are the kind of models I think we can consider. With on-demand, as I mentioned before, this is event-driven, so you really need to have the PrEP available to you. There's a wonderful program in New York City that includes the Play Sure Kit. That's a great video on YouTube if you haven't seen it, where people, and it's a much definitely on the lines of a sexy presentation about how to have PrEP or other prevention methods with you. But whichever way you go, really good counseling for both adherence and understanding risk is necessary to make the prevention effective adherence concept work. And then I will start to conclude by talking about a little bit of a perspective on adherence in prevention. And I think these concepts have already been introduced, so I'll go through it fairly quickly. But I just wanted to review, as you've heard, that the first few studies about PrEP didn't come out and say that if you take it, it works, right? We know that initially there was some efficacy, but adherence was an issue. And over time, adherence has increased as people have gotten more of a better understanding of what really is efficacy, not necessarily with a placebo-controlled design, and getting to choose to take it, coming forth and saying, I want PrEP. And those people who want PrEP are pretty good at taking it. So let's think about the vaginal ring in this context. As you've already heard, the early 2 studies showed fairly modest efficacy, and the adherence values I'm showing here are based on drug levels within the blood, which is really something that doesn't give us a great idea about adherence over time. Again, it's the periodic formulation, so we'd like to know how is adherence over the time that it's meant to be used. And with the drug levels, you may only get a few hours before detection, so it's hard to say. If you look at the residual drug in the ring, that gives you a better idea. And what you can see here, at least with the Aspire trial, efficacy was much higher when you looked at women who had evidence of drug within the ring. So even though it's a lower number, you see higher efficacy. And we can ask Dr. Nelb about equivalent data from the ring study, but I imagine it's similar. And we'll see as Hope and Dream come out to find out exactly how well women are able to do when these issues that are presented here over to the side about having more knowledge of efficacy and not having a placebo-controlled trial and being able to come forth and say, yes, I want this. How good is their adherence? And we'll see what the true effectiveness is in that situation. And then a final point, we would all love to have a perfect formulation of PrEP that everyone takes and that works perfectly. But in reality, we really just need to find what is going to work for individuals. And even if that formulation is not 100% efficacious, if people are using it more, they'll get more benefit than a formulation that may be more efficacious but not working for them as an individual. So really marrying those 2 concepts is important. So in conclusion, adherence and acceptability are multifaceted. There are many things to consider. Choice is incredibly important, and one size certainly will not fit all for an individual or for their choices and situations and contexts over their lifetimes. Multipurpose technologies are really promising for acceptability and adherence. I really would like to emphasize this concept of prevention, effective adherence, and shifting to a way of thinking about adherence when it's necessary, and acknowledging that there are times when people shouldn't be taking PrEP because they really probably don't need it all the time. And then to think about adherence and effectiveness over time as PrEP is evolving. So thank you very much for your attention.
That was great. Okay, you have to listen to one more set of slides, and then you get to ask questions. So just to wrap up, it was great to see this paper from Zina Stein in that last, because we are in an amazing place now where we actually have stuff to talk about. And you can spend an entire 6 hours today thinking about PrEP from 7 a.m., where there were 2 sessions that worked on PrEP and microbicides at the same time, and looking forward to an amazing plenary from Sheena, and now this session today. Then you get to go to lunch. We heard from Angela about right drugs, right sites, right concentrations, and right length of time with fancy figures. We heard from Ian. This is very memorable. This does not hold just for rectal microbicides. I think this is all things PrEP, including the things that are in development now, are still somewhat more on this right side of the screen than on the left side of the screen, and we have to keep working on that. We heard about the Dapivirine ring and the congratulations on the EMA filing in last month, and looking forward to long-acting and multi-purpose prevention. And then we thought a lot about adherence and how it is important, and how it is somewhat obvious that you can't get efficacy if something is unused. And we thought, and several people mentioned their titles today. The 2 of us, Rafi and I, we were not given the title of this session either. We were given the assignment to be here. So this topical agent's as good as systemic PrEP may have some tone to it, we decided. But as good as on several levels, right? Because it's as good as on the biologic level, it's as good as for an individual and what they want, and it's as good as for public health. And this quote that Jessica pulled out of Zina's paper reminds us that something that may not be as well, that may be not as good biologically, but may be better used, may have an important role in public health prevention and preventing transmission at the population level. And so when we think about all of these prevention tools that are coming, it's figuring out the range of options that will work for an individual and that will have the greatest public health impact. So I encourage you now to ask questions. Otherwise, I will give you that look that Rafi had in the photo of me at the beginning. So please come to the microphones, and we'll have questions now. Thank you, everybody, and thank you to our speakers. Ken.
Hi, Ken Mayer from U.S. Great panel. Jared, you had at the very end you mentioned metronidazole. I'd just like the panel to talk a little more about altering microflora, what you see the role of colonizing lactobacilli that could deliver products, and also products particularly for rectal microbicides that might also be active against bacterial STIs since they're so prevalent these days.
When do you want to address that?
Yes, thanks. So I think that the first issue is that we're having to better understand which are the organisms that do impact on topical formulations of PrEP. And it seems at this point that organisms such as Gardnerella, Prevotella, Mobilingus, those are the organisms that seem to be impacting at least tenofovir levels. These studies are underway, and so we'll get a better sense of how they impact on a range of other antiretrovirals as well. So there's really two things that we need to be thinking about. The one is, how do we get rid of these organisms that are negatively impacting drug levels? And so for that, we're really thinking about antibiotics. And in the case of most of these organisms, Flagyl is an effective agent. But even when we do use antibiotics like Flagyl, or metronidazole, we still have the issue of, if you treat it, do you cure it? Does it go away forever, or does it come back? And what we've seen, certainly from our experience and from others, is that when you do treat with metronidazole, you have the problem that it recurs. So that's leading to a whole exciting new range of research that's being done, led by a company called Ocell, which have developed, are looking at, in clinical trials, using a lactobacillus in a capsule as a replacement. So they are trying to colonize the vagina with the favorable organism. And so you're really looking at two sides of the same coin. So you use metronidazole to suppress the not-so-good organisms, and you make available lactobacilli in this capsule form to see if it can colonize the vagina and make available all the good organisms. Now, these are still at a very early stage, so we don't know, how long they last and whether they have the positive effects we'd like to see, but that's where the field is going. And I'd be very happy to have Sharon comment. She's really one of the pioneers in this area.
Hello, and thank you for a very interesting symposium today. I had a comment and then a couple of questions. So the comment comes from a session yesterday, some of you may have attended, on the future of PrEP, and a remark that Ambassador Birx made, that the epidemic has changed, but the research has not, and the researcher and the product developers haven't changed. We're still in the sort of emergency mode, try to find something that works, and we haven't yet understood that user wants something not only that works, but that is pleasant, comfortable, beautiful, and easy to use. Part of the issue, I think, with the topical versus systemic is the issue of side effects. And we know, for example, in contraception, that it's a huge barrier to use. I haven't heard much about this today, and I'd love to hear from the panelists about side effects and the pros, potential pros of a topical application versus systemic application. The other question is more directly to Angela, is we still struggling about where the drug needs to be in terms of the mucosa versus the lymph nodes. And can you talk a little bit more about the pros and cons regarding topical versus systemic absorption for that, in that respect? Thank you.
Who wants to talk about side effects and usability? Anneline?
I can talk from the Dapivirine vaginal ring perspective that because there's extremely low systemic absorption, you're looking at pico levels, and meaning we haven't seen any systemic side effects with the Dapivirine ring, as well as looking at the local side effects. They were minimum between a normal population versus placebo versus your active ring, with no impact on pH, BV, STIs, and that range of side effects. But the important for the ring is, as I emphasized, is that it will be a novice product in the Africa setting. So what we've seen during the clinical trials, that first 2 months of knowledge sharing, of coaching, and help women to build their trust in the ring is actually critical. Up to the point, because initially when they look at the ring, they perceive it as maybe big, maybe not easy to insert, up to the moment where with the first, second ring, that they've had the confidence to use it. The other important part is that they need to build the confidence in themselves that their male partner might not feel it, because there were reports of male partners that felt it. And I think those are aspects that we really need to think through during our implementation and demonstration process. But the biggest advantage, there were no systemic side effects.
Probably worth briefly mentioning long-acting injectable and rectal. I can talk to both of those to some extent. Long-acting injectable, the main adverse issue is injection site reaction, pain, discomfort, and so forth. And depending on which population you look at and what questions you ask, the prevalence varies, but it's always quite high. I think the Eclair study that Marty Markowitz wrote up in Lancet HIV a year or so ago is very illuminating in that regard. I think the prevalence of grade 2, which is moderate injection site reaction, was about 50%. Grade 3 was actually 20% in that population. But when they were asked, would you use this or recommend it to your friends, 87% said they would. So that raises big questions, I think, for people like yourself about acceptability and how you ask questions and so forth. Rectal microbicides, predictably, the adverse event profile is generally benign and diarrhea-related GI distress, but not a major issue. I mean, others could talk to oral PrEP, but I think oral PrEP's really generally very well tolerated. There may be an induction period of a little bit of nausea or GI distress, but that goes. So, I think that every product has its issues. But Angela, I'd love to hear Angela's comments about this mucosa lymph node issue, because it is becoming, both in terms of pathogenesis and PK, an issue. So.
Yeah. So if you look at Tom Hope's data, he does not see any trafficking going on. It's all it's all infection in the stroma, and so I think if you can target the stroma and you can stop infection there, I don't think you need to worry so much about regional lymph nodes, and I think that's why there has been efficacy with topical products. I think we'd see a lot worse outcomes if we actually needed to get to regional lymph nodes. Now, if the concentrations are low, there's an oral product where the concentrations are low in the stroma, then I think, yes, absolutely, you need to have a drug at regional lymph nodes. So I think the study with TAF will be very interesting because those diphosphate concentrations are quite low in mucosal tissues. We're still trying to understand exactly why, but they could be very high in regional lymph nodes. So I think you could probably target either site, but you need to have high concentrations at one of those sites in order to prevent infection.
It is ultimately quite amazing that we have a topical product at all. It teaches us much about where it could be. I think also just to speak a little bit more to the side effect question that was raised, is when we think about, we don't have a lot of information yet in sustained use of any of these products. And when people are thinking about systemic use of something for a year or two or more, if people are going to want to have sustained use, and sustained use across their life cycle, women who are pregnant or breastfeeding or other things. So it's a good for us to all keep in mind. Oh,
Hey, thank you.
it comes to you, but only you.
So thank you very much, the panel, outstanding panel, and a lot of stalwarts also sitting there on the front row. The question goes perhaps to Slim because he raised the issue, but probably there are other people here in the audience. Adam, and I don't know, Nikki's here, Sharon. What you mentioned two important cofactors of determining efficacy. One being the microbiome and the other inflammation. But you base the very elegant studies that you publish on tenofovir gel and Caprisa. What do we know about other agents and whether this is a mechanism that we have to take into consideration even when developing other agents for topical application?
So I'll answer to the extent I can. So what we showed when you are looking at the topical formulation of tenofovir, that the actual drug levels in the lumen can fall drastically even within the first 4 hours. And in fact, Nikki's work showed that the levels reduced to about half the concentration within 4 hours. So what we're seeing is a very rapid metabolism of tenofovir in the presence of Gardnerella. The question then is big. So what effect would that have if you were not delivering the tenofovir topically? What if you were delivering it systemically? And the evidence we have from one study done by Connie's group looking at partners PrEP is looking at the microbiome, what effect that had on efficacy, and they found no effect. So it suggests that the microbiome may not be directly relevant to systemic PrEP. But I think we need to expand those studies. We need to have a better understanding of the microbiome in detail because the Partners PrEP study didn't look at the microbiome in the same way as the other studies did. So we do need to look at the microbiome in a much more comprehensive way, looking at the range of organisms and what impact each of them is having. And to the extent to which these organisms impact on the potential drug levels in the stromal layer of the lumen, that's what we need to answer. And I think the answer to that question is it probably is not going to be an issue in systemic PrEP, but we don't have a definitive answer on that just at this time.
And what about other topical agents, Adam?
Yeah, Dapivirine, and Ian, I have a question for you about rectal microbicides, and what are your thoughts on rectal microbicides and the microbiome interactions?
So I thought you were going to answer a question.
Sorry. Lots of questions.
Someone answer.
You're asking me about, what was the question?
What is the impact of microbiome and Gardnerella on other
drugs? Well, my lab is specifically looking at interactions between Dapivirine and bacteria, but Nikki Klatt's lab is looking at interactions between other drugs and the microbiome, and she's finding some very interesting things happening, and I think she's presenting that tomorrow, but I don't know. And if she were here, I could pass off the mic, but I think that it's definitely an unanswered question, and it needs to be explored further. And Ian, could you answer, provide some
rectal
discussion
microbicides in the microbiome? In all our phase 1 studies conducted within the MTN, we've always taken the opportunity to do semi-quantitative assessment of microbiological flora, culture-based techniques, obviously. and never seen anything really very significant. Okay, so that's at a very superficial level. Clearly the microbiome plays a critical role in many GI conditions and diseases. There's no doubt about the importance of that. What we've also done in those phase 1 studies, as you know, along the way, is an intensive compartmental PK and PD. And generally speaking, when the model is valid, which is another issue, come on Wednesday to hear about it, shameless self-promotion, the explant model, you see very good PK-PD correlation. So if the microbiome is doing something, it's certainly not obvious. But those studies are limited in size and scope, and they haven't been paired with the kind of state-of-the-art molecular characterization of microbiome. So what we do know from the Barcelona group, who's represented here by Pep Coy, is that within gay men, and gay men with and without HIV, you see rectal dysbiosis. You do see profoundly different microflora patterns. And so there's a basis for concern. I think we'd need to do more focused studies to see if that actually correlates with actually differential compartmental PK.
For Dapivirine, I would just call people's attention to an abstract for tomorrow. Sharon Hillier, who might be here still, will be presenting on microbiome and plasma and genital concentrations of Dapivirine. So that's an 11 a.m. session tomorrow.
All right. Thank you. That was a really great presentation. I have a two-part question. One is going beyond the microbiome. Can you hear me?
Yeah.
Okay.
Yeah.
At CROI a couple of years ago, they found in real-world settings, both in San Francisco and in New York, an upswing in STI, syphilis, gonorrhea rates in PrEP clinics. So what's the net efficacy, not just inflammation, but inflammation from STIs, like in the context of PrEP, with net efficacy, with incident syphilis or incident gonorrhea? And also, my second part was, we're seeing a lot more, there breakthrough infections with resistant people who acquire it from people who have resistant HIV infections. So with an increased use of integrase inhibitors in the past 7 years, how are you projecting potential resistance data for cabotegravir and dapivirine, as well as tenofovir?
And Lane, do you want to talk
maybe
about it? Yeah, we could go.
Just to comment briefly, one of the challenges of trying to interpret the existing PrEP studies, both topical and systemic oral PrEP, is that within these studies, there's a high level of STD care that's provided. So in other words, you diagnose somebody and you treat them either syndromically or diagnostically. So to do a subgroup analysis within these PrEP studies is not an easy task to answer your question, because we've asked the same question in writing the guidelines as to, would PrEP be effective in somebody who has gonorrhea? And we actually don't know the answer definitively. The existing trials don't suggest that that's a problem, but we can't answer it definitively.
I think from the Dapivirine rings are all I can just add on what Slim said. We saw STIs was higher in your younger women at baseline, but it was not identified as a covariant throughout the trial. But once again, the trials were not powered to look at each individual covariant, but STIs were not one of them to being picked up.
And I think Sheena, who's sitting at the back of the audience, would tell you in the PROUD study that, what was it, Sheena, about 35% prevalence of rectal bacterial STIs, chlamydia or gonorrhea, and really the only breakthroughs, of which there were very few in that study, were linked to potentially non-adherence, not bacterial STIs. So. Your other question about resistance is probably a topic for a whole session. I'm not sure we'd want to take that on just in the last 5 minutes, but obviously it's something, as these products are rolled out, we have to continue to monitor.
I think so, we've got time, Craig. Yeah.
Yeah. Hi. So I'm Craig Cohen, the University of California, San Francisco. I just wanted to give an update to what Slim mentioned around Lactin V and OCEL, and back to Ken's question. We're actually quite advanced now. We're in the middle of a phase 2B trial, which is being conducted in the U.S., supported by the NIH STAR STI Clinical Trials Group. We're also, so that we plan to be fully enrolled sometime around January 2018 228 women randomized 2-to-1 to Lactin-V versus placebo. These are treating women with BV. We're treating the women for 12 weeks with Lactin-V or placebo and looking at the difference in BV recurrence. So watch this space in about another year, a little bit more than a year, we'll have the follow-up data. And then we're moving along with completing the pre-IND or the preclinical testing of their second generation product, which is called Mucocept CVN, or genetically modified Lactobacillus jensenii, where Lactin-V is a crispata. So I just wanted to give you that update information. We're actually further along than what was mentioned earlier.
Thank you.
Thank you. I'm Charlotte Watts from DFID. A really great panel. I really appreciated the reference to Zena's comment that in the end, the level of protection an individual gets is not only the efficacy of the product, but the consistency with which it's used. But in public health terms, also the other factor is coverage. And can we achieve sufficient coverage of the groups that are most vulnerable to HIV? So I'd like to hear from any panelist about what's the critical thing when we think about scalability and coverage that we should be paying attention to linked to the product pipeline. Thank you.
I like that question so much. I think, can we start at the end and just march through the whole panel and have them answer this? Because that's what really we're all here for, right? Is to achieve coverage and impact. So, from your perspective.
I think I would take two perspectives on that. I mean, for coverage, the individual has to choose to take it up, so offering effective choices, I think, is going to be very critical in making those available to people in ways that make sense to them, so a very patient or individual-centered approach. And then this concept of prevention, effective adherence, I think could make, from a public health perspective, make PrEP seem a lot less scary in terms of the amount of investment in finding ways to integrate PrEP into time-limited periods in people's lives that could be co-developed with other public health sources, so it's not a standalone, lifelong commitment. I think that could really help with getting it out there from the public health side.
I mean, I think I mentioned in my talk, and I'd add again, I think we've reached a very good point where we now have oral PrEP. You know, we have a vaginal ring, hopefully soon to be licensed, and we have an injectable on the horizon. And I think the coverage, by which I mean programmatic engagement to develop other product lines, is probably diminishing. But maybe that diminishing situation should be compensated for by increasing work on getting coverage. I mean, just like treatment, for the prevention, we have oral PrEP. You've seen the figures. It's not everywhere. It should be. Why isn't it? So I think those drivers are going to be both financial, as you know, but also behavioral and community engagement and other issues. So I think we really need to focus on what we have and really make sure people are able to access it. I would still love to argue for other formulations. People need choices, but I think we live in a resource-limited situation. Where should you prioritize getting coverage or continuing to develop new products?
Yeah. This is Ramesh Akina from Colorado State University. I just have a general question to the panel about HIV-2. HIV-2 also seems to be spreading in some parts of the world. Are there any studies looking specifically on this aspect for topical or oral PrEP?
I think specifically no, although certainly tenofovir is active against HIV-2 and has been, there have been a couple of demonstration projects done in West Africa. Dapivirine would not be, and was specifically not evaluated in an HIV-2 endemic area, but tenofovir should be.
Yeah, that probably becomes an issue, especially when you're considering combination, agents. Some agents may not work on HIV-2. Just a general comment.
Thank you.
Other panelists, we're at our last minute, so final comments on coverage impact.
If I can just to make one observation for Charlotte, I've been struck over the years at the different levels of motivation we see for the use of oral PrEP or any form of PrEP among MSM than we see in the other populations, particularly in the general population of high-risk young women. And everything that I've seen is the level of motivation, the freedom it gives them in how they engage in sex and so on, creates a set of motivations that leads, in my view, to a much higher demand for PrEP in that group. However, among young women, which is the large part of what I deal with in the general community, who are young, mobile, somewhat carefree, you have the challenge that they don't see themselves at risk to start with. And so demand creation for any form of PrEP is going to be a really big challenge. And to get coverage is going to be a really big issue. And I think that that's a challenge that's got to be taken up, because we're talking about healthy people, people who are not sick, who don't interact with the healthcare infrastructure. And so we have to find ways to capture them in the community. We have to find ways in which to highlight to them the risk of HIV, and then we've got to highlight to them we have a potential solution to it. Then we've got to highlight to them they have to take a tablet every day. So you can see where this is going. This is a big challenge, and certainly from our demonstration project, we see all of these challenges in a very substantial way. So to me, it's a very different situation talking about PrEP in MSM and talking about PrEP in young women. I mean, we're talking about a group that's really hard to reach.
Coming from that point, being engaged with many young women forums over the past weeks, I think the critical one will be to listen basically to them and say, how do we optimize our current options within the young women sector, but also to say, how do we engage with them to make our environment where they can seek prevention options and treatment user-friendly for them. I think that was one of the key messages that came out, and I think it can link to Ian's packaging slide, is that the fear to go and attend a clinic. How will I be treated? Will it be actually coming back to me and stigmatize me? So I think it's a complex package that we really need to overcome in order to be successful, especially in our young women, as well as in our public sector, keeping in mind we need to engage in a public sector that's overworked, that's stretched, that's very often been blamed for under-deliver, but there's also not supply. So it's really to engage with government, political leaders, to change that mindset in order to make a difference. But for me, the key is to optimize what we have, and when you're looking at product development, is how do you complement your current options. But our key will be our young women and how to really engage with them, empower them to take ownership of their wealth, and not just HIV, but overall well.
Right. With that, I'd like to thank the organizers of this conference and this symposium, particularly Jean-Michel Molina, for inviting us all here today. I'd like to thank our incredible panelists for a great discussion, and to thank all of you for amazing questions and a fantastic discussion. So thanks very much.